Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 13 from our Campbell's 12th edition biology textbook. This chapter covers meiosis, so let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
All right, welcome back. So again, Chapter 13, meiosis. So we're going to be talking about the formation of the gamete cells, the sperm and the egg. So recall that genes are present on chromosomes. Genes typically code for proteins, and these genes are the units of heredity that are made up of segments of DNA. Heredity is the transmission of traits from one generation to the next. This is called inheritance or heredity. We are studying how our genes are passed on to the next generation. The first step of that being meiosis, the formation of what are called the gametes. The gametes are the reproductive cells. These include the sperm and the eggs. And your non-reproductive cells are called your somatic cells. These are the rest of the cells in your body. So your body, you can think of it as your body is made up of two main types of cells. Your somatic cells, which make up pretty much your entire body, and then your gamete cells. These are only the sperm or the eggs. Does that make sense? So somatic cells, that's your skin cells, your brain cells, your heart cells, your liver cells, and then the gamete cells, which are only referred to the sperm and the eggs. And in this chapter, we are learning about meiosis, and that is the cell division that only occurs in the reproductive organs, the ovaries or the testes, to form the gamete cells, the sperm and the eggs. So again, the only purpose of meiosis is the formation of the sperm and the eggs. So the only place meiosis occurs is in your reproductive organs. Now, do you remember from chapter 12 when we were talking about the cell cycle and mitosis? I told you that humans have 46 total chromosomes, and this is because you inherit 23 from the egg and 23 from the sperm. So you essentially inherit a set of 23 from the egg and a set of 23 from the sperm. So take a look here. This is how it works. The sperm cell contains only 23 chromosomes. So it contains a set of chromosomes, 1 through 23. Same with the egg. The egg contains a set of chromosomes, 1 through 23. And when the sperm fertilizes the egg, now you have your two sets of chromosomes. You have 46 total chromosomes. You have a set of 23 from your paternal lineage, your father, and you have a set of 23 chromosomes from your maternal lineage, your mother. And remember, these are known as homologs or homologous chromosomes. And so we have 46 chromosomes. Now, any cell with homologous chromosomes inside, any cell that has the set from mom and the set from dad, any cell that has both sets from both parents is called a diploid cell. So the fertilized egg is called a zygote, and because it has two homologous sets of chromosomes inside, this is known as a diploid zygote. Diploid meaning two homologous sets of chromosomes inside of the cell. Now let me ask you this, can you beat Wicket? Are the sperm and the egg diploid? Do they have two homologous sets of chromosomes inside? A set from mom and a set from dad? Oh, that's right, Wicket. You can see here that the sperm only has 23 chromosomes, and the egg only has 23 chromosomes. And I'm going to show you how the sperm and the egg got those chromosomes and why they only have 23 chromosomes. That's the process of meiosis. That's what we're going to be discussing today, is how the sperm and the egg formed. But you should know that these are not called diploid cells. These are called haploid cells. So this is the haploid sperm. This is the haploid egg. And please remember that the egg and the sperm are called the gamete cells. These are the sex cells. Or when the sperm fertilizes the egg, this is known as the zygote. And it's a diploid cell now because you have two homologous sets of chromosomes. So your body, your somatic cells that make up your body, those skin cells, brain cells, heart cells, liver cells, these cells are all diploid cells. And the haploid cells in your body are simply the sperm and the egg. Again, each pair of homologous chromosomes includes one chromosome from each parent. The 46 chromosomes in a human somatic cell are two sets of 23. One from the mother, one from the father. Remember, this is what makes up a diploid cell. Now, I also want you to know that instead of writing diploid cell, you can shorten that to 2N. 2N means a diploid cell. You have two homologous sets of chromosomes. So if you just see N by itself, that's a haploid cell. It has one set of chromosomes. But if you see 2N, that refers to a diploid cell with the two homologous sets from each parent, or one set from each parent. So for humans, the diploid number is 46 total chromosomes, and that's 2N. You have two sets, which makes up your 46 chromosomes. And again, it's only the reproductive organs, the ovaries and the testes, that produce the haploid gametes. The gametes are the only type of human cells produced by meiosis rather than mitosis. We learned about mitosis in chapter 12, the previous chapter. We learned all the phases of mitosis and everything. When the cells of your body divide, those somatic cells that make up the majority of your body divide, they divide by mitosis. But specifically in the reproductive organs, this is where meiosis occurs, and that's only to form the sperm and the egg, the sex cells. And as you saw with the sperm and the egg in that previous figure, meiosis results in one set of chromosomes in each gamete. That means that the sperm and the egg contain a total of 23 chromosomes. One set. All right, now one thing I want you to understand is that sperm cells themselves do not divide. So one sperm cell does not become two sperm cells. And egg cells do not divide either. One egg does not become two eggs. So where do these sperm and these eggs come from? Well, they come from special diploid cells that are in the reproductive organs. Diploid meaning they have 46 chromosomes, they have two homologous sets of chromosomes, but they're special. These are the cells that divide to give rise to the sperm and the egg. Does that make sense? So these cells have a special name. They're known as the germ cells, and they are the cells that undergo meiosis and result in the sperm and the egg. And you can only find these germ cells in the reproductive organs. In the reproductive organs, you have those germ cells with 46 chromosomes. Those germ cells undergo interphase. Do you remember interphase? We talked about that in the previous chapter. If you don't recall interphase, make sure to go back to chapter 12 because we discussed that during interphase. This includes the subphases G1, where the cell grows, S phase, where the DNA is replicated. So if you started with 46 chromosomes, and then during interphase, S phase, those chromosomes are replicated, how many chromosomes would the germ cell have now? That's right, Wicket. 92 92 chromosomes, right? Your 46 chromosomes get replicated. So now that germ cell possesses 92 chromosomes. And then remember, following S phase is G2 phase. This is the phase where the cell continues to grow, but it also replicates the centrioles and the centrosomes. And now you're all good to go. You're ready for the process of meiosis. Now, what's really interesting about meiosis is that there isn't just one round of cell division, like mitosis. You remember what happened in mitosis? You had interphase, and then you had mitosis, which was, that one cell became two. Well, here, there's actually 2 rounds of cell division. Does that make sense? So your diploid germ cell divides one time by a process known as Meiosis I, which has its own prophase, its own metaphase, its own anaphase, and its own telophase. These should look familiar, right, from mitosis. But then that results in how many cells? That results in 2 cells, right? Well, those 2 cells divide again, okay? Those 2 cells divide again by a process known as Meiosis II, which has its own prophase called Prophase II and its own metaphase, anaphase, and telophase. So let me ask you this. If you started with one cell, one germ cell in the reproductive organs, and the cell divided twice, how many cells would you end up with? That's right, Wicket. 4 cells. You'd have 4 cells, and those are known as the gamete cells, okay? Those are your gamete cells, the sperm and the egg, okay? Now, let me ask you this. If you started with a germ cell with 46 chromosomes, that germ cell copied all 46 chromosomes during S phase of interphase, and that resulted in a germ cell with 92 chromosomes, okay? Well, then remember, a cell with 92 chromosomes divides once by meiosis I to give 2 cells with 46 chromosomes, and then each of those cells divides again to give 4 cells with how many chromosomes? 23. Now, does it make sense why the sperm and the egg have 23 chromosomes and not 46 like a typical somatic cell? Well, it's because the DNA was only copied once, but the cell divided twice. Does that make sense? So what I want you to know is that there's only one interphase where the DNA is replicated, and that occurs before meiosis I. There is no interphase between meiosis I and meiosis II. And because the DNA is only replicated once, but the cell divides twice, you're going to have gamete cells, the sex cells, the sperm and the egg, with only 23 chromosomes per cell. I hope that makes sense. We're going to go into more detail now. But then once the DNA has been copied and the cell has a copy of all the chromosomes, we are ready for meiosis I, prophase I. So let's go in to see what really occurs during this prophase I step of meiosis I. So here is prophase I. I want you to look inside and see, do you spot any difference here between prophase I of meiosis and prophase of mitosis from the previous chapter? I just want you to take a quick look here. It looks like the centrioles have already been replicated from G2. That's great. They start moving to opposite ends of the cell. It looks like the nucleus is intact, and that's great. That's what you would expect, right? During prophase, the nucleus should be intact. And inside, we see our chromosomes. But there's something strange about these chromosomes. I just want you to take a look at these strange chromosomes. It looks like, do you guys remember how the difference between homologous chromosomes and sister chromatid pairs? Remember in chapter 12, I said it's very important that you remember the difference between the two, because that makes these chapters so easy if you can just keep that clear in your mind. So what I'm going to do is I'm going to hop to the board real quick, and I'm going to show you again quickly the difference between sister chromatid pairs and homologous chromosomes. And then I'll meet you back here so we can make better sense of what's happening during prophase I. Don't forget that before meiosis, special germline cells in your testes or ovaries, your reproductive organs, undergo interphase. If you don't remember interphase, quick review. During interphase, your diploid cell with 46 chromosomes undergoes a G1 or cellular growth. The cell grows in size. Notice that you have homologous chromosomes inside. You have one pair of centrioles. You then have subphase S, where the chromosomes get replicated. Now you have 2 of every chromosome. You have 92 total chromosomes. At this point, you have what are known as sister chromatids. Remember, homologous chromosomes are the maternal and paternal set, right? Or maternal and paternal version of every chromosome. Sister chromatids, on the other hand, are identical copies of the same chromosome. So at the end of S phase, you have sister chromatid pairs forming. And then in G2, which is the final subphase of interphase, the centrioles replicate. The centrioles replicate, and they start moving to opposite poles of the cell. Okay, the nucleus is still there. The DNA starts to condense, but it's not quite condensed. All right, so that's where we leave interphase behind. So don't forget, interphase has just happened. We are now ready for what's called meiosis I. During prophase I, tetrads form and crossing over happens. What does that mean? If you look inside the nucleus, the sister chromatids pair up with homologous sister chromatids as a tetrad. What that means is both of mom's chromosome 1 attached to both of dad's chromosome 1 with a synaptonemal complex holding them together as a tetrad. So this would be the tetrad for chromosome 1, for example. This would be the tetrad for chromosome 2. And notice what's happened here. It's almost like the tips have exchanged between one of the 2 sister chromatids here and one of the 2 sister chromatids there. They've swapped genetic information. That's called crossing over. Crossing over has happened. Okay, so now you have 1, 2, 3, 4 chromosomes that are no longer at all identical. Crossing over increases genetic variability. Crossing over results in more genetic variability, okay? None of these 4 chromosomes are the same after crossing over. So again, what's happening during this prophase I, don't forget each cell has 92 total chromosomes. Tetrads formed, crossing over happened, okay? And crossing over increased genetic variability. We're now ready for metaphase I. So welcome back from the board. I hope you noticed that at prophase I of meiosis, some strange stuff is happening with the chromosomes. If you remember, during mitosis, it was those sister chromatids that formed, right? So during prophase of mitosis, sister chromatid pairs formed. However, if you noticed from my board presentation just a second ago, something more complex is going on during prophase I of meiosis, and that is this. These structures are forming. They actually consist of 4 chromosomes, and these are known as tetrad structures. And I'm going to show you exactly what a tetrad structure is, because it's these tetrads that form during prophase I. So here is a tetrad structure. A tetrad is when a pair of sister chromatids joins up with the homologous pair of sister chromatids to form a tetrad structure. Tet means 4, and this is a fitting name because this is 4 chromosomes all hanging out together. So, for example, let me give you an example. If these were 2 exact copies of dad's chromosome 1, this would be the 2 exact copies of mom's chromosome 1 underneath. Does that make sense? That's what homologous chromosomes mean. The two blue ones, for example, are the paternal homologue, and the two red ones are the maternal homologue. The two blue ones are identical to each other. The two red ones are identical to each other. So the two blue ones are sister chromatids. The two red ones are sister chromatids, but they're binding together. And it's this synaptonemal complex that holds the sisters together as homologous pairs. Now, here, this is known as a tetrad structure. So what you need to know is that during prophase I of meiosis, tetrads form. And the reason they form is really interesting. At this point, one of these blue sisters is going to exchange genetic information with one of these red sisters. So there's going to be a break. Imagine if I have scissors. Imagine if I could cut this blue tail right here and then cut the red tail at the same spot, okay? Imagine if I could cut this tail and cut the tail right below and swap these out, swap the two out so that I have the tip of the red tail up here and the tip of the blue tail down there. This is a process known as crossing over. So this is what happens. First, tetrads form, and then there is this crossing over event that happens. You see this crossing over happens, and look what the result of crossing over is. Look at this. After crossing over has happened, and remember, crossing over only happens in one of the blue tails with one of the red tails. The other sister does not undergo crossing over, okay? So if that's true, let me ask you this. After crossing over, are any of the 4 chromosomes identical anymore? Because before, look, the two blue ones were identical and the two red ones were identical, right? But after crossing over, are any of these chromosomes identical? No, that's right, Wicket. No, they're not, right? Because of crossing over, none of these 4 chromosomes are identical anymore. And that's great because that is exactly what the purpose of meiosis is. The purpose of meiosis is to increase what's known as genetic variability, having more genetic shuffling going on, because this is part and parcel with sexual reproduction. During sexual reproduction, the genes are shuffled around, the chromosomes are shuffled around as much as possible because this gives the offspring the best chance at surviving, right? This leads to what's known as adaptation. So the purpose of crossing over is to increase genetic variability. Isn't that neat? So this is exactly what's going on during prophase I of meiosis. So what do you need to know? During prophase I of meiosis, tetrads form and crossing over happens. So let's move on to metaphase I. Take a look at metaphase I. During metaphase I, it's those tetrads that line up at the center of the cell. Remember that imaginary plate called the metaphase plate? So during mitosis, it was the sister chromatid pairs that lined up at the center of the cell. But during metaphase I of meiosis, it's the tetrads that line up at the center of the cell. And remember, those tetrads have undergone crossing over, so the little tips have been swapped over. And it's these tetrads that line up down the center of the cell. But there's a little more to it than that. So let me give you a little more detail about what's going on and exactly how they're lining up at the center of the cell, because that's important to understand. All right, so at this point, a human cell in metaphase I would have 23 tetrads. That's how many tetrads would be found in a human cell, which is in metaphase I. However, just for illustration, this here shows a cell with 3 tetrads, okay? Only 3 tetrads. So let's call this chromosome 1, chromosome 2, and chromosome 3. So again, remember the tetrads are, you know, they all share the same chromosome number. What does that mean? So for example, if this is chromosome 1 from dad, that's an exact copy of chromosome 1 from dad. And then if this is chromosome 1 from mom, that's an exact copy of chromosome 1 from dad, and that's a tetrad. And by the way, I don't know why, but they have not drawn the crossing over. But remember, this has undergone crossing over. And if this was chromosome 2, this would be 2 copies of chromosome 2 from dad, and this would be 2 copies of chromosome 2 from mom, 2 copies of chromosome 3 from dad, 2 copies of chromosome 3 from mom. So again, there's 3 tetrads in this cell, and this is a cell in metaphase I. Now, what's really interesting is that there's a reason why they drew this cell in 8 different ways, right? And if you notice, if you take a close look, you'll notice that the reason they drew 8 different versions of this cell in metaphase I is because the tetrads are lined up in different orientations. So what do I mean by that? Look at this. For example, take a look at this. Here, for chromosome 1, the blue ones, the paternal sister chromatid pairs are on the left, and the maternal sister chromatid pairs are on the right. However, did you know that it's equally likely that they would have lined up the other way, with the maternal homologs on the left, with the paternal homologs on the right? So what I'm trying to say is there's an equal probability of the chromosomes lining up with this way, with the paternal homologs on the left, as they are lining up the other way with the paternal homologs on the right. And that's not just true for chromosome 1, that's also true for chromosome 2. See here with chromosome 2, you have the paternal homologs on the left, whereas here you can see the paternal homologs on the right. And chromosome 3, here the paternal homologs on the left, but in this cell, the paternal homologs on the right. So what that means is that not only do the tetrads line up at the center of the cell during metaphase I, they also line up completely randomly, with each tetrad having an even chance, an equal chance of lining up with the paternal homologue on the left or the paternal homologue on the right. So this means that there are many different combos. There's many different combinations of chromosomes that can line up this way. In fact, to figure out exactly how many, you would take you would take 2 and rise it to the power of the number of tetrads you have. So for example, here we have 3 tetrads. So to understand how many different combos of arrangements we could form during metaphase I, you would do 2 to the power of 3, which is 8. And that's why 8 different pictures are drawn here. Humans have 2 to the power of 23, right? So that's a huge number. That's over 8 million. Okay, so there are many different ways that your tetrads can line up down the center of the cell. Isn't that really neat? And that does what? Remember, that increases genetic variability. And this ability for the tetrads to line up randomly, giving you such genetic variability, is known as independent assortment. Why? Because these tetrads assort independently of one another. Each has a 50-50 chance of having the paternal homologs on one side or the other. All right, that about sums up what happens during metaphase I, but now we're ready for anaphase I. Remember, anaphase, when we talked about mitosis, it was the sister chromatids that separated from one another, right? But here, during anaphase I of meiosis, we have tetrads, and it's the maternal homologs that separate from the paternal homologs. So what does that mean? You remember that synaptonemal complex that was holding these this whole complex in place, the tetrads together? Well, that synaptonemal complex breaks down, and the chromosomes separate, and the two red ones go in one direction, the two blue ones go in another direction. Let me show you how that looks. Again, during Anaphase I, what's happening? It looks like the tetrads have broken apart such that the paternal homologs, that means both copies of dad's chromosome 1, for instance, separates from the maternal homologs, which is both copies of mom's chromosome 1. These would be known as sister chromatids, but because of this little blue tail due to crossing over, these would technically be called non-identical sister chromatids because they're not fully identical anymore, though they once were. And then on the other side, these are also known as non-identical sister chromatid pairs, right? So again, the same thing happens with chromosome 2. Let's say this is chromosome 2. Both of dad's chromosome 2 go to this new cell. Both of mom's chromosome 2 go to this new cell. So what's happening is, you could say it this way. During Anaphase I, the homologs separate. That means blue separates from red. Paternal separates from maternal. Another way of saying the exact same thing is that this pair of non-identical sister chromatids, or the paternal pair of non-identical sister chromatids, separates from the maternal pair of non-identical sister chromatids. That's a fancy way of saying the blue ones separate from the red ones. At this point, those chromosomes move towards opposite ends of the cell, and you're ready for telophase I. During telophase I, you have the daughter nuclei starting to form. So a nucleus starts to form around this DNA, and a nucleus starts to form around this DNA. Now I want you to ask you a question, and that is, if you take a close look at this, would you say that these two are genetically identical cells? You know, kind of like, remember at the end of mitosis, after cell division, you had two genetically identical cells. Well, take a close look at these two cells, and let's see if you can beat Wicket and answer the question, are these two cells genetically identical? That's right. As always, no, these cells are not genetically identical. There's two red ones in this cell. You know, like for example, look at this cell on the bottom here. This cell has 2 copies of mom's chromosome 1, but 0 copies of dad's chromosome 1. Both copies of dad's chromosome 1 went to this cell. So, and also this cell has 2 copies of dad's chromosome 2. You see that? So there's no way these cells are identical, because this cell has both copies of mom's chromosome 1, be it that one is a non-identical. These are non-identical sister chromatids, but they're still technically sister chromatids. So there's 2 copies of mom's chromosome 1, 0 copies of dad's chromosome 1. So guess what? That means that these cells are anything but identical. This one has both copies of mom's chromosome 1. This one has both copies of dad's chromosome 1. This one has both copies of dad's chromosome 2, and this one has both copies of mom's chromosome 2. So what I'm trying to get across is that by the end of telophase I, by the end of meiosis I, this results in 2 cells that are not genetically identical. And this is something else that really confuses students, and that is that even though both of these cells would have 46 chromosomes at the end of meiosis I, each of these cells has 46 chromosomes, so they technically have 2 sets of chromosomes, right? Would this qualify as 2 diploid cells? And let's see if you can beat Wicket. Yes, right. This does not qualify as 2 genetically identical cells, and it does not qualify as 2 diploid cells. And look what it says here in my notes here. By the end of meiosis I, 2 genetically unique, that means different, haploid cells have formed. And I know this is very confusing for students because the students say, well, there's 46 chromosomes in this cell and there's 46 chromosomes in that cell. So that's 2 sets. So that's diploid, right? Well, that's not quite right. And I'll explain why. Here, do we have 2 copies of chromosome 1? Yes. But do we have a mom and a dad chromosome 1? Like, for example, so essentially, do we have homologous chromosome 1s? Do we have a mom chromosome 1 and a dad chromosome 1, or just 2 copies of mom's chromosome 1? That's right. We only have 2 copies of mom's chromosome 1. Okay, that would be cheating to call this a diploid cell. You can't just have 2 copies of almost the exact same thing and call yourself diploid. Does that make sense? You guys follow me on this? So you can't just have twice the number of the same exact chromosomes and call that 2 sets of chromosomes and call yourself diploid. A diploid cell, not only does it have 2 sets of chromosomes, it has 2 sets of homologous chromosomes, which means 1 set is from your mom, 1 set is from your dad. This cell obviously does not have that. It does not have 1 set from mom and 1 set from dad. This is 2 copies of mom's chromosome 1. There's 0 copies of dad's chromosome 1. Both copies of dad's chromosome 1 are over here, right? And then this cell has 2 copies of dad's chromosome 2 with 0 copies of mom's chromosome 1. Both copies of mom's chromosome 1 went to the other cell. So yes, technically, these 2 cells have 46 chromosomes, 2 sets of chromosomes. But because there aren't technically homologous chromosomes in each of these cells, just 2 copies of the same information, that's cheating. That doesn't qualify as 2 diploid cells. So by the end of meiosis I, this results in 2 genetically unique haploid cells. And because of this, meiosis I is often referred to as what's called reduction division, because the ploidy of the cells reduced from diploid 2N to haploid 1N. Okay, now we are entering meiosis II, and this begins with Prophase II. During Prophase II, remember where we left off, we had 2 cells, but I'm just going to pick up with one of the 2 cells. But remember, at the end of meiosis I, we had 2 cells. But I'm just going to show you what's happening in one of those 2 cells. But obviously, know that there's double everything in your mind, right? So remember what we had? We had a cell that's haploid. Even though it has 46 chromosomes, it's haploid because it has 2 of mom's chromosome 1, 2 of dad's chromosome 2, etc., etc. And remember, these are technically non-identical because crossing over had happened. All right, so what do I need you to know? These are still technically sister chromatid pairs. So sister chromatid pairs form. That's what I need you to know. During Prophase II, sister chromatid pairs form. And if you want to be extra specific, yes, you could say that non-identical sister chromatid pairs form. Okay, that's what's happening during Prophase II. Now, in Metaphase II, those sisters, those non-identical sisters line up. Okay, the non-identical sister chromatids line up down the metaphase plate. Anaphase II, you guessed it, the sisters separate from one another. The non-identical sister chromatids separate from one another. So for example, 1 copy of mom's chromosome 1 is headed to this cell. 1 copy of mom's chromosome 1 is headed to the other cell. And now for Telophase II. Now, during Telophase II, daughter nuclei form. And let me ask you this. How many total cells would be present at the end of Telophase II? That's right, it would be 4 cells. Because remember, during meiosis I, 1 germ cell became 2 haploid cells. By the end of Telophase II, those two cells divide again to form 4 gamete cells, right? So at this point, you have 4 genetically non-identical gamete cells. Because look at this. Let me ask you this. Is this cell identical to this cell? Take a close look. No, because look, this mom's chromosome 1 has a part of the paternal homologue. It has a part of dad's information on it, whereas this mom's chromosome 1 is just mom's chromosome 1. Does that make sense? So because of crossing over, these two cells are not identical. And if you're curious, because of independent assortment, these two are not going to be identical to the other two. So what that means is at the end of Telophase II, at the end of Meiosis II, this results in 4 genetically non-identical gamete cells. These are the sperm and the egg. Okay, does that make sense? None of these 4 sperm, none of these eggs have the same genetic material. They are all genetically different. They are all genetically distinct. And that's wonderful because that's exactly what the purpose of meiosis is. The more the genes are shuffled around, the better the offspring have at a chance to adapt and to survive, right? Isn't that neat? And let me ask you this. How many chromosomes total are in each of these gamete cells at this point? Can you beat Wicket? That's right. 23 chromosomes per gamete. So each sperm cell will contain 23 chromosomes. Each egg would contain 23 chromosomes. So yes, you have one set of chromosomes in each of the gamete cells. And because, let me tell you this, because during meiosis II, the cell goes from having 46 total chromosomes to only 23 chromosomes per cell, meiosis II is termed equational division. Equational division. So now let me ask you this. This is important, and I may ask it on the exam for my class. Which meiosis, is it meiosis I or meiosis II, that most resembles mitosis? What do you think? That's right, Wicket. It's actually meiosis II. And I'll tell you why. Let me explain why. First of all, let's look at meiosis I. Remember during prophase I, what did the DNA form? Remember it formed tetrads and crossing over happened? That's all new concepts, isn't it? Did any of that stuff happen during mitosis? The answer is no. And then remember during Metaphase I, those tetrads lined up randomly by independent assortment? That's also a no. That none of that happened during mitosis. Anaphase, the homologs separated, that didn't happen in mitosis. And so, however, look at meiosis II. During meiosis II, prophase II, sister chromatid pairs form. Yes, they're non-identical, but sister chromatid pairs form. Isn't that what happened during mitosis? Yes. Then in Metaphase II, it was those sister chromatid pairs that line up down the center of the cell. Isn't that what happened during mitosis? Yes. Anaphase, sister chromatids separate from one another. That's the same as mitosis. Telophase, those sister chromatids move to opposite ends and form new daughter nuclei. So what I want you to understand is meiosis II is much more similar to mitosis than meiosis I is. And why? Well, because, you know, tetrads never formed during mitosis, crossing over never happened, independent assortment. Meiosis I is its own beast. It's its own thing. So if I were to ask you which one's more similar to mitosis, it would have to be meiosis II. Again, mitosis, which was covered in chapter 12, mitosis occurs in the somatic cells of your body. The purpose of mitosis was to conserve the number of chromosome sets, producing 2 cells that are genetically identical to the parent cell. However, in meiosis, which was the focus of this chapter, chapter 13, involves reduction in the number of chromosome sets from 2 diploid or 2N to 1 haploid or just simply N, producing 4 cells that differ genetically from each other and from the parent cell. And these are known as the haploid gamete cells, the sperm and the egg. There were 3 events unique to meiosis, and all 3 occurred in meiosis I. Now, this include synapsis and crossing over, synapsis being when the tetrads formed, and crossing over, which increased genetic variability. Remember that? Then the alignment of the homologue pairs at the metaphase plate with independent assortment, which also increased genetic variability. And then the separation of those homologs. Remember, the homologs are what separates during Anaphase I, which is also unique to meiosis as well. Now let's take a quick break time with Gizmo and Wicket and see what these 2 little guys are up to, and we'll be right back to finish off this chapter. Welcome back from break time with Gizmo and Wicket. Let's carry on. I have some interesting discussion here about meiosis, and it has to do with the genetic variability that occurs during meiosis. Remember that during meiosis, this is responsible for the variation that arises in each generation. 3 mechanisms contribute to genetic variation. Remember, crossing over, which occurred during Prophase I of meiosis, independent assortment, which occurred during Metaphase I of meiosis, and then lastly, random fertilization, right? Because it's a random sperm that fertilizes a random egg. So all 3 of these actually contribute to genetic variability and genetic variation. Remember what I said earlier during Metaphase I, that you can figure out how many variations there could be during Metaphase I by taking 2 to the power of the number of different chromosomes there are. So for humans, we have 23 different chromosomes. So 2 to the power of 23 is over 8 million different combinations of chromosomes. That means that each person can form 8 million different combinations of chromosomes in their gametes, in the sperm or the eggs. And that's true not just for the father, for instance, but the mother as well. So the father can form 8 million different types of sperm based on their chromosomes, and the mother can form 8 million different types of egg based on their chromosomes. And so for a child to, come to be, a 1 in 8 million sperm would have to fertilize a 1 in 8 million egg, right? So isn't that rare? Wouldn't that result in an ultra rare combination, an ultra unique combination in the offspring? Isn't that really interesting to think about? And so what they say is this random fertilization of a random 1 in 8 million sperm variation with a 1 in 8 million egg variation adds to the genetic variation because any sperm could fuse with any ovum or unfertilized egg. The fusion of 2 gametes, each with 8.4 million possible chromosome combinations from independent assortment, produces, get this everyone, produces a zygote with any of about 70 trillion diploid combinations. So think about that. You were born, right? And you were a, combination of your genetic material from both parents, right? And that was 1 in 8.4 million sperm fertilizing a 1 in 8.4 million combination egg. So the odds of having you were 1 in 70 trillion. Isn't that fascinating to think about? And I just want to point out that this 1 in 70 trillion has to do with just independent assortment and random fertilization. Does it count? Does it factor in crossing over? Does this 70 trillion figure count crossing over? No, crossing over adds even more variation. If you added the crossing over variation, it would be in the quadrillions in the amount of different types of sperm and egg and combinations that are possible. It would be just astronomically high amount of variation if you think about it that way, because crossing over can doesn't necessarily happen the same way twice. You can have single crossover events, double crossover events, triple crossovers. There's so many different ways that crossing over could have happened. So, this 70 trillion just refers to independent assortment and random fertilization. So what am I trying to get at? I just want to do a thought experiment for you. Like, imagine, so your parents had you, right? But what would be the odds of your parents having you again, right? So imagine you were born with your genetics. So it's possible for your parents to have a child with your genetics because you were born, right? But what would be the odds of your parents, I don't know, like in 2 years down the road, having another child and that child just randomly having your exact genetics, right? Is it possible? Yes. Is it probable? No way, not really, right? It's like really, really, really not probable. It's probably so improbable that it's probably never, ever happened in the history of humanity. Isn't that interesting? So, for a couple to have a child with particular genetics and to later on have a child with that exact same genetics, it would be so astronomically rare that it would be like winning the mega jackpot lottery several times in a row, I'm betting, so it would just not happen. Now, invariably, students would ask me, well, what about identical twins? What about identical twins? Yes, it's true that identical twins have the exact same DNA. But do you know why? It's because identical twins started out as one total sperm and one total egg. So it really was one sperm fertilizing one egg, and that led to a diploid zygote. That zygote started to divide and divide and divide, and at some point, a little part of that zygote, a little part of that blastocyst popped off and divided as though nothing happened, and that resulted in two people. Isn't that interesting? So the reason why twins have the exact same genetics is because they didn't come from two sperm and two eggs. Those twins started out as one sperm and one egg. So they were the ultimate roommates at one point, if you think about it. They literally were roommates. And the room was a cell called the zygote. Isn't that interesting? So if you know identical twins, or if you are an identical twin, just think about that for a second. You and your twin were in the same exact cell at one point, and you came from one sperm and one egg. Isn't that interesting? And that's why you have identical DNA to one another. But the odds of you guys having identical DNA, but you were born at different times with different sperm and different eggs, would be so astronomically small that it's probably never, ever happened. And so you know what they say? This is a funny one, everyone. They say that you are incredibly, incredibly unique, just like everyone else, right? So let's finish off. So with that, I just wanted to get back to the board, and I want to do the board talk with you one more time because it just, it just nails all the most important steps, all the most important aspects of every phase of meiosis. So let's get back to the board, and then we'll call it a chapter. So don't forget, interphase has just happened. We are now ready for what's called Meiosis I. During Prophase I, tetrads form and crossing over happens. What does that mean? If you look inside the nucleus, the sister chromatids pair up with homologous sister chromatids as a tetrad. What that means is both of mom's chromosome 1 attached to both of dad's chromosome 1 with a synaptonemal complex holding them together as a tetrad. So this would be the tetrad for chromosome 1, for example. This would be the tetrad for chromosome 2. And notice what's happened here. It's almost like the tips have exchanged between one of the two sister chromatids here and one of the two sister chromatids there. They've swapped genetic information. That's called crossing over. Crossing over has happened. Okay, so now you have 4 chromosomes that are no longer at all identical. Crossing over increases genetic variability. Crossing over results in more genetic variability. Okay, none of these 4 chromosomes are the same after crossing over. So again, what's happening during this prophase 1? Don't forget, each cell has 92 total chromosomes. Tetrads formed, crossing over happened. Okay, and crossing over increased genetic variability. We're now ready for Metaphase I, where tetrads line up by independent assortment. Okay, notice that the tetrads have lined up down the center of the cell along this imaginary plate called the metaphase plate. Okay, tetrads line up. And what does that mean, independent assortment? Independent assortment means each tetrad has a 50-50 chance of lining up this way or the other way. See the way the first tetrad lined up? It lined up with the blue chromosomes on the left, or the paternal homologs on the left. Well, that one could have very well, that tetrad could have lined up the other way with the blue ones on the right. And so could the second one. The blue ones are on the right, but it could have very well 50-50 chance lined up the other way. That's what independent assortment means, and that leads to a lot of genetic variability. Okay, that leads to over 8 million different ways that our tetrads could line up, because we have 23 of these tetrads during this phase, not just 2 like this simple picture here. All right, so then at the next phase here, Anaphase I, the DNA separates. But what's separating from what? Okay, we say that the homologs separate. What does that mean? This pair of sister chromatids separates from the homologous pair of sister chromatids. In layman's terms, these two, dad's chromosome 1, separate from these two, mom's chromosome 1. So all of dad's information for chromosome 1 is headed to the cell on the left. All of mom's information for chromosome 1 is headed to the cell on the right. And if we're looking at chromosome 2, it's the opposite story. All of mom's information for chromosome 2 is headed to the left. All of dad's information for chromosome 2 is headed to the right. So this is going to result in 2 genetically different cells, right? So telophase I, you see, you form 2 cells, and those 2 are considered haploid. Let me get to this in a second. First of all, notice that daughter nuclei form. Daughter nuclei form. Each cell now has 46 chromosomes. Okay? But realize this. Look, in this cell, you have 2 dad's chromosome 1s and no mom's chromosome 1s. In the other cell, you have 2 mom's chromosome 1s and no dad's chromosome 1. This means that even though you have 46 chromosomes, which means 2 sets of chromosomes, it's not quite diploid anymore. It's not quite diploid anymore. These cells are technically haploid because you are missing in this cell a mom's chromosome 1. You only have 2 of dad's chromosome 1. If you do not have homologous chromosomes inside of a cell, that cell is not technically diploid. Does that make sense? So even though, yes, technically this cell has 2 sets of chromosomes and this cell has 2 sets of chromosomes, because they're not homologous sets of chromosomes, that's not diploid. So for in layman's terms, because you don't have a dad's chromosome 1 and a mom's chromosome 1 to back it up, you just have 2 of dad's chromosome 1, that's not diploid, that's haploid. And because of that, meiosis I is considered reduction division, which means, what did you do? You went from 2N diploid cell, 2N diploid cell, which has homologous chromosomes, mom and dad information for every chromosome, to N, 46 chromosomes, and haploid, because you no longer have mom and dad homologous information in each cell. Okay, so moving on, I'm just going to show you meiosis II. I'm just going to show you what's happening in this cell. I'm not going to worry about that cell, but just know that whatever's happening in this cell is happening in that cell. I'll show you what's happening in this cell here in meiosis II time. All right, this is meiosis II, prophase II. And notice what's going on here. This cell has 46 chromosomes. Even though it's N, it's haploid because it's lacking homologous chromosomes. During this step, sister chromatids form and condense. You see this sister chromatid pair forms. For example, this would be 2 of dad's chromosome 1. This sister chromatid pair forms. For example, that would be 2 of mom's chromosome 2. Okay, and you're done with prophase II. Next you have metaphase II. What's lining up down the center of the cell? It looks like the sister chromatid pairs line up at the center of the cell. And I wrote that here. The sister chromatid pairs line up. Anaphase II. What's separating from what? It looks like those cohesin proteins holding the sisters together broke, and the sister chromatids separate from one another. So the sister chromatids separate from one another. All right, sister chromatids separate from one another. Now, lastly, I'm showing you here what happened to all of the cells. So not just what happened to the left cell here, but also what's happening to the right. So I'm showing you all 4 of the resultant cells. Each cell now has 23 chromosomes. These are now called the gamete cells. Daughter nuclei form, okay? And what you should realize is none of these 4 cells is genetically identical anymore. These are all 4 genetically non-identical cells. So for example, this cell and this cell are not genetically identical. And you know why? Because crossing over. You see this dad's chromosome 1 is just dad's chromosome 1. This one has a bit of mom's chromosome 1 in it because of crossing over. So this cell and this cell are not the same due to crossing over. Neither are this cell and this cell. You see, these cells are not the same due to crossing over. This is mom's chromosome 1. This is mom's chromosome 1 with the blue tip. So these are not the same. These are not the same. And then these 2 are not the same as these 2. You see, these 2 up here are not the same as these 2 down here because independent assortment. Remember during Metaphase I, independent assortment happened? So that's why these 2 and these 2 are not the same. Okay? So none of these 4 cells are the same genetically. All of these cells are now haploid. They have 1 set of chromosomes, and these are called the gamete cells. And meiosis II is considered what's known as equational division. Okay, this process of equational division. Why? Because you've simply gone from 46 chromosomes to 23. Whereas remember, meiosis I was called reduction division. Why? Because you've gone from a diploid cell to 2 haploid cells. You've reduced the ploidy of the cells. And one last thing you should understand about meiosis, and that is that it looks like meiosis II is a lot more similar to mitosis than meiosis I. Okay? And look, meiosis II, sister chromatid pairs form, sister chromatid pairs line up, and sister chromatids separate from one another. Well, that's pretty much what was happening during mitosis over there. Whereas in meiosis I, tetrads form, tetrads line up. Oh, tetrads formed and did crossing over, which is different. Tetrads lined up with independent assortment, and then homologs separated. That's completely different than what happened during mitosis over there. So I hope this helps. This is my rundown of meiosis.
Okay, welcome back. Hopefully this was helpful. Please let me know in the comments below if you have any questions or if it made sense. I appreciate you tuning in, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.